Physics felt very mature. Biology was expanding in all directions. Felt like there was more to discover in biology.
Dr. Stephen Quake is a pioneering bioengineer and one of the founding fathers of microfluidics. He's also known for developing key technology for noninvasive prenatal testing.
Huge red herring to try to separate maternal from fetal DNA. You don't need to do that.
A serial entrepreneur and former Howard Hughes investigator, he most recently served as the head of science at the Chan Zuckerberg Initiative.
When things get mature, I get bored.
You're right. In our conversation today, we'll explore his journey in transforming the landscape of biotechnology, the challenges of bridging science and industry, and his vision for the future of health and personalized medicine. Welcome to Stanford Department of Medicine's Inside Look at the Future of Medicine. Well, Steve Quake, hard to know where to start with you and your career and the impact that you've had, but we always love with our guests on the show to talk about their background and, you know, what first drew them into science and medicine. And I've talked to you about this before, so I know one or two of the stories that relate to that. But tell us for the listening audience, how, you know, when did you first know you were going to be a mad scientist, and how did you move on from there?
The first experiment I might have done as a young boy was to try to make gunpowder.
Right. And what led you to, how old were you first of all?
Oh my gosh, I don't know, somewhere in elementary school.
Yeah.
My parents had given me this 2-volume set of books called The Way Things Work.
Okay.
And you could read through it and explain like all kinds of technological things and how this and that works.
Yeah.
And,
Yeah.
and I like shooting off fireworks.
Yeah. Who doesn't?
Exactly. Unfortunately, I still have all my fingers. Yeah. But I thought, I should try to make my own. And I read in the book, you know, the formula for gunpowder and what goes in it. And so I went and tried to gather the ingredients and mix them up. And, you know, didn't quite appreciate the role of compression and all. But so it was a failed experiment.
I see. That's good that it was a failed experiment. You told your parents you were doing this?
Of course not.
No,
of course not.
That's not right. Yeah. In the backyard.
Yeah,
Yes,
exactly.
Right.
Could have burned down the forest or something.
Yeah, yeah. So building things, inventing things, cooking up new things was something, sounds like it was, you know, almost in your DNA, as it were, from an early stage.
Absolutely. And, you know, I grew up in the early days of the personal computer revolution. And so that was in the mix as well. I learned how to program the computer and use it to control things and learn a little bit of electronics and that sort of thing as well.
You used to get the magazine. I mean, I think I did the same. We sort of shared this. We would get a magazine through and it would have software code written on it and you type it in and then hack it a little.
Absolutely.
That was also your experience.
For sure.
Yeah.
For sure. All those trade rags coming from this far off place called Silicon Valley.
Yeah. For me, it was a very far off.
It felt far off for me. I was upstate New York, so it was far off for me too.
You know, when did that transition then to an idea of a speciality called science, or, you know, a profession called science?
You know, when I first went to college as an undergraduate, I knew I was interested in physics, but I was also interested in engineering computers. And so I was sort of undifferentiated. It was probably going to be one path or the other, but I had some very inspiring science, physics and math teachers.
And where was your undergrad?
Here at Stanford. Yeah. And a couple of very inspiring teachers. And that sort of set me on the path of science and kind of stuck with it.
Yeah, it seems like it's gone pretty well. But computer science and engineering, obviously today, you know, very, very obvious the impact that they're having.
Yeah, maybe I took the wrong path.
I don't know. But biology is obviously an area where you've had, you know, a huge impact. Biology and medicine will come to talk about that impact. But you started on the engineering side, sort of making things work. How did you first start to pay more attention to biology, or what was it drew you into that?
Yeah, so as an undergraduate, I trained in physics and math and was very much in that mode. But when I got to graduate school and I was trying to decide what to specialize in for my PhD, and I spent my whole first year trying to survey all of physics about, you know, what would be the most interesting problems to work on. What I got to was, well, the interface between physics and biology would be the most interesting because physics felt very mature as a field and biology was expanding in all directions, and it just felt like there was more to discover in biology using the sort of approaches and philosophies of physics. And so I was at that interface of biology and physics for a long time, but then brought in this ideas of measurement and developing new measurement apparatuses, and that's where the engineering started to fold into it.
Right. I remember also in one of our prior conversations, you talked about the impact of reading Feynman's work. Talk about that a little bit.
Yeah. Oh, when I was in high school, I had his autobiography and stumbled across it and read it, and just loved his fearlessness and his irreverence.
Yeah.
That was very inspiring to me. I mean, I reread it again last year, and it certainly didn't age well.
No, for sure. That's for sure.
It didn't age so well, but for the moment and of the time, it was.
60s. When would he have written that?
I think he wrote it in the 80s.
Oh, I see. Okay,
but it was definitely about things from World War II era. Beyond.
Exactly.
Yeah,
right. No, very interesting. And of course, your own path took you, geographically at least, down to Southern California, of course. But you were at graduate school, and graduate school was started here, right? But you ended up in Oxford.
I did my doctorate in theoretical physics at Oxford. But I spent part of it here visiting to do experiments.
I see. Yeah. And why Oxford?
For the adventure of it, basically. I had not had the opportunity to study abroad as an undergraduate, and so I thought, well, I'm either going to travel abroad and work as a ski bum or something, or if I get a fellowship, maybe I'll go study abroad. I ended up getting a fellowship, and so that paid for my experience.
Nice. So that was a Marshall Scholarship.
Yeah.
And you went to Oxford, pretty prestigious thing. Remind me what college you were at?
Merton.
Ah, Merton. Yeah. One of the older ones.
The oldest.
Yeah. Okay. So, but pretty amazing, but very different place from Stanford and New York.
Oh yeah, I struggled with that.
Yeah,
I struggled with that. There were formal dinners every night.
Right.
You had to wear a tie and a jacket, an academic gown.
Don't think I've ever seen you wearing a tie.
Yeah.
I have seen you wearing a jacket. Yeah.
So, I did all kinds of experiments to understand what the true limits of the dress code were.
I see. Okay.
And, you definitely needed to wear the tie. You had to have a collar on your shirt. But I got through with a rugby shirt.
Okay.
Socks ended up being important. They didn't like it when I didn't wear socks one day. But the big discovery was that they could regulate the dress code, but they couldn't regulate taste.
I see.
So I had the most hideous polyester ties you could imagine, and those were fine.
Yeah, okay.
Like they didn't give me a hard time about it. But that was
any
sort of
color, any fabric.
I mean, day glow. Yeah. It was wild.
Right. Right. Physics professor for sure in the making.
Oh.
Right. And then you came back, and then you were at Caltech.
Yeah, I started my faculty career at Caltech. Spent almost a decade there.
Yeah.
Started as assistant professor and moved up the ranks, and it was a wonderful, wonderful place and experience. I mean, just the history, the focus on what it does. I mean, they really just do great science, and kind of the community there was just terrific for me because I was able to really go across disciplinary boundaries. I sort of collaborated with a ton of people.
Yeah.
Knew almost everybody, and which is a great spirit of working together.
Yeah. Did you have a specific mentor down there that sort of led you in the direction of measurement, and it sort of helped underline your interest in biology and the intersection with physics, or was it really around collaboration?
There were so many things that were going on in that period. I mean, I worked very closely with a fellow named Axel Scherer. That's where we got the microfluidics off the ground. He's sort of E-beam lithography person, double E background. And just a lovely person. And we mentored several students and postdocs together and launched a whole bunch of the microfluidics work there.
Yeah.
Frances Arnold was a mentor for me.
Yeah.
And we worked together on using those microfluidic tools for screening and molecular evolution. And she's remained a good friend to this day.
Yeah.
And those are probably the two closest collaborators. But I worked with a bunch of the biologists. I mean, Henry Lester, Bruce Hay, Mel Simon. It just kind of went on and on. Yeah.
Well, I mean, many people have been exposed to and been impacted by your inventions that came sort of in your Stanford time and beyond. Some of them, especially the doctors, might not know that really your early career was around essentially inventing microfluidics.
Mm-hmm.
The principles were there, but really in terms of making it practical and making applications. That's the first time I'm not even sure if we met, but the first time I was in a talk that you gave was in Seattle at the Institute of Systems Biology. Mm-hmm. And it was all about microfluidics. So that's how we all knew you. And of course, Fluidigm was a huge success and really has continues to impact the field. Maybe just for the audience, just remind us what that is. And I mean, there are valves that are, you don't call them these, but other people call them Quake valves.
They do.
Tell us about that.
Yeah. So, when I started at Caltech, I was interested in trying to do automation of biology because I knew I wanted to work on the interface of physics and biology. I knew the biologists tended to have labs with lots of people and there's a lot of manual labor involved, and that's not what I wanted to mentor. And so I thought, all right, let's build tools that automate biology. And I had been following, these ideas about trying to make the integrated circuit of biology, miniaturized plumbing with valves and pumps and pipes and all that. And it started to dabble in it a little bit when I was a postdoc, and really then leaned into it as an assistant professor. And we tried a bunch of different things. And eventually, one of the things we landed on was these valves, where we figured out how to batch fabricate tens of thousands of valves on a single chip, sort of arbitrary plumbing complexity. And then, went about trying to figure out what to use that for. And one of the first things we decided to use it for was protein crystallization. And I knew we were onto something when I sent my postdoc up to James Berger's, or my student, up to James Berger's lab for a week and at Berkeley, and he did in a week more experiments than James's best postdoc had done in a year.
I see.
So we were like, okay, we're onto something.
Right.
Yeah, exactly. And then it just went from there.
Yeah. And then Fluidigm, was that your first company?
Yeah.
So tell us about that. Was it just a very obvious thing to do? Was it something you'd always wanted to do, or did it come out of somewhere else?
Well, it was a little bit maybe in the family history. My dad was an early software entrepreneur, so I'd seen over his career him founding companies and getting them off the ground. And it was also a bit that it was pretty clear that making these chips was complicated enough that no biologist would be able to do it. It required a bunch of specialized knowledge. And so to see these tools have impact in biology would require somebody making them in some kind of commercial availability. And so, I didn't want to stop with the work just being published as a paper. I wanted to have very broad impact. And it seemed like starting a company was the way to do that.
Yeah. Did you find it easy? It's never easy.
It's never easy.
But,
oh my gosh.
Yeah, raising money is never easy. One of the things that I think places like Caltech, something like Stanford stand out is that there's quite a few people around who've done it. So you can sort of, round a corner and find somebody who can give you advice. Was that what you found at Caltech, or were you really trying to break out on your own and sort of had to learn it by yourself?
Much different environment at Caltech. And Caltech had a tech transfer office that was run by a fellow named Larry Gilbert in those days, and he was terrific. And he mentored me and many other faculty on how to be an entrepreneur. I eventually found the CEO, Gaius Worthington, who was up here in the Bay Area. And we founded Fluidigm, and he quit his day job, and he convinced two other guys to quit their day job, very confident they'd be able to raise money and launch this thing. And like almost a year later, still hadn't been able to raise money. They're living on ramen. One guy was getting ready to sell his motorcycle. It was much harder than any of us realized it was going to be. And biotech was in kind of a down cycle then. And Larry introduced us to, one of the things he did was introduce faculty entrepreneurs to potential investors. He eventually introduced us to a fellow named Bruce Burroughs, who became the first investor in the company. And Bruce wrote us our first check, and that got the whole thing going. And then it was,
off and running.
Yeah.
When was the first, do you remember how long it was between when you started the company and when the first kind of product really shipped to the first customer?
Oh my gosh. That was, let me think about that. It must have been 3 or 4 years.
Yeah.
Because we had to figure out how to do manufacturing. And that in itself was a challenge.
Because you spoke about this during your grand rounds, but universities are great at some things, and they're great environments for collaboration and for pushing envelope, demonstrating, the N of 1. But then to scale, manufacturing or to even think about selling something is a whole different set of skills.
Oh.
Last time I checked, which was last night, but correct me if I'm wrong, you have started 12 companies.
Yeah.
Give or take. So we'll definitely touch on 1 or 2 of them as we move forward. But there are probably, when I first contacted the Office of Technology and Licensing here at Stanford, we were talking about a variety of different faculty who'd started companies also. And they, I think they refer to the Quake factory, because in the sense that you were a factory of company formation. But each of those has a very specific idea and is often based around a very specific technology that is new. It's an amazing adventure. Maybe we can come to talk more about that sort of duality as a faculty member. But yeah, I really wanted to get to some of what I would say in terms of lives affected, millions of lives affected, in this case patients, and many of the listeners I think here will be familiar with non-invasive prenatal testing. And they're really, I think, there are several people who contributed to early work, but I think from my perspective, there's really two big names in the world, and you both worked both together and, you know, maybe even a little competitively for a while. Dennis Lowe, of course, in Hong Kong, and your work here at Stanford. That all started though with something quite close to home.
Yeah, I mean, so I never worked on anything clinical before. It was all very basic science oriented, and when I first became a father, we had to interact with doctors and have amnio and this sort of thing. And it was just, you realize just this was not a good state of affairs to have to take risks about your baby just to get a diagnostic measurement. And so that sensitized me to it, that there's an important problem to be solved there. And it was in the back of my mind for a number of years. And then eventually I stumbled across this literature on cell-free DNA. And of course, in the late 90s, Dennis and Jim Wainscoat had been first to show that with molecular methods that some of the cell-free DNA in the blood is from the baby.
Yeah, and in their case, they were focused on the Y
chromosome.
So,
exactly.
And so picking up in the context of a male baby, women, of course, say female mother, and the Y chromosomes must be coming from the fetus.
And that work had set off a decade-long effort to try to build a practical diagnostic. And many things were tried and failed, and there's no biochemical difference between fetal and maternal DNA and such forth. No way to enrich.
And but Dennis was in Oxford around that kind of time. Did you overlap with him?
Have you ever seen the picture?
No, I have not.
I'll show you the picture sometime.
Yeah.
So it turns out we were in Oxford at exactly the same time.
You were? Wow.
He was getting his medical training. I was doing my PhD.
Yeah.
His wife was a physicist in my department, in the physics department, and we graduated at the same time. Right. We never met, but at Oxford as students. But at the graduation, someone snapped a picture of him and her, and I'm in it.
Wow.
We were standing near each other.
Really?
Didn't realize it. And the first time I visited Dennis in Hong Kong, he says, I'm going to show you something.
Oh.
He goes, pulls out this picture, and it's three of us in the same picture wearing graduation robes at Oxford.
Know that.
Yeah, I have a copy of it somewhere.
Okay, amazing. Amazing.
Yeah, but so anyways, there was this long effort to try to figure out how to take advantage of this phenomenon. And I think the key discovery was this counting principle. That if you can count the molecules and measure overrepresentation, that was the breakthrough that enabled all of NIPT, and that's sort of what we did here at Stanford.
And this is counting specific molecules of DNA that then map to specific chromosomes. Mm-hmm. So you could look for overrepresentation of chromosomes and therefore see.
Yeah, it was a huge red herring to try to separate maternal from fetal DNA. You don't need to do that.
Right.
All you need to do is look at the overrepresentation. And it has this amazing property that it can be as sensitive and precise as you want if you just count high enough. The higher you count, the better it works. It goes like 1/√n. Yeah. So it's a very unusual property for a diagnostic test.
And what technology were you using?
So we started trying to do a digital PCR.
Right.
And Christina,
just remind us what that is.
Okay. So digital PCR is a way to get around the nonlinearities of PCR amplification by trying to amplify single molecules in discrete wells. Yeah. And it had been around for a long time as a concept. Yeah. It goes back to someone named Sykes. And Bert Vogelstein and others had been trying to do with emulsions and to publish some papers
for cancer.
Right. And we'd seen that. And I had a rotation student in the lab who became my PhD student, Christina Fan. And I said, for your rotation project, why don't you try to replicate this Vogelstein paper and let's try and make digital PCR happen. Mm-hmm. And she couldn't do it. Yeah. It's a very fickle thing. The chemistry is challenging. The emulsions are hard to work with. And so then Fluidigm had just then made their digital PCR chip, and they made the first commercial digital PCR product. Okay. So then I said, all right, let's use that. And that worked right away. Yeah. And so we were able to count molecules. We could do these kind of dummied up experiments and show that it would work in principle. And then we tried to do it with the real clinical samples, and you couldn't count high enough on a single chip for those. And I said, I don't care. Use as many chips as you need. Let's spread the sample over them. It doesn't matter what it costs. Yeah. And she didn't listen to me. She was very conscious of my budget, which I guess I
We
appreciate it.
appreciate that.
But I wanted her to do this experiment.
Yeah.
And so eventually the first sequencers had just come out then.
Yeah.
We had ours at Helicos, Illumina had theirs. And I said, all right, fine. If you're not going to do it with digital PCR, let's do it with sequencing and try that. That's another way of counting.
Yeah.
And for some reason, she agreed to do that.
Okay.
And that's how we ended up doing the counting. And that's mostly how it's done today.
Yeah. Yeah. Remarkable. And then as a test, you've obviously talked about amniocentesis, which was the principal method for getting a sample of fetal DNA in the past, or the cells, in order to do karyotyping. But in terms of impact, in terms of the number of millions of lives affected, I think you made this point well in your talk. If you're giving an individual a choice between a large needle that would be approached towards the head of their unborn child or a blood test, most people are going to choose the blood test.
Absolutely.
And of course, it's a different test in the sense that it's for screening, but it essentially takes you to the same place, which is an answer to a question.
It's interesting, this question of screening versus diagnostic.
Yeah.
And, my perspective on that, when a diagnostic test doesn't work well, it gets used as a screen.
Yeah.
If it works well, it's a diagnostic.
Yeah.
And in the NIPT world, there are some that don't work very well because, they're optimized for cost or something like that, or just weren't designed well. Yeah. And so, they all market them as screens, but the best ones are as good as amnio, in fact. Yeah. And so, I think it is just a matter of time and accumulation of data and experience before that actually becomes the gold standard.
Yeah, I think the test characteristic is what matters, not what you label the test as. You also showed the paper I remember when it came out, the way you sequenced essentially an entire baby's genome from the blood of, with some knowledge of the family, but otherwise from the blood of the mother and cell-free DNA, which is remarkable.
Yeah.
Yeah.
Yeah. I mean, so this counting principle works not just for chromosomes, but for, however you want to bin the genome. You could do structural variation. You made the bins so small, they're just a single base pair.
Yeah.
That lets you read out essentially the whole genome. And in that paper you're talking about, we did it two ways. So one way was to get haplotype information from the parents, and then you could reconstruct the fetal genome from that. The other was to do it completely naive to the parents. And so you could just draw the blood sample without having worked out the genetics of the mom or the dad, and do a pull down to get the exomes and do the counting principle on the exomes. And that worked as well.
Yeah. And I mean, it's essentially, it has taken over for all the obvious reasons. I mean, the test characteristics have improved over time. More than just chromosome counting has happened over time. It's certainly routine sequencing of fetal genome isn't something that happens yet, but obviously the technology moves in that direction, and it's become an everyday routine clinical test that everyone really expects to have. It's not always reimbursed. It seems to be reimbursed more often, thankfully, than it used to be. Things take a while, it turns out, in diagnostics. And then the company you started was taken over by Illumina in that case. And of course, now this is just a routine medical test that happens millions of times every year.
Yeah. It's been an amazing journey.
It's an incredible journey in terms of real
marked
impact.
A real transition in the diagnostic world, I think, in that from the business perspective, people used to look down on it. Venture capitalists didn't like it because diagnostics were viewed as antibody tests, $10, $20, something like that.
Yeah.
And so hard to build a big business around it. But, invasive biopsies have been expensive. Those are thousands of dollars a test. Yeah. So if you can replace that with something that's safer and better, you can charge less than what the invasive costs, but still enough that the business starts to look very, very attractive and the numbers add up. And Well,
when you think about the current state of non-invasive testing, or the prior state of non-invasive testing, it was based on, one protein marker, ultrasound of the neck of the baby. It just, that speaks to a world that now sounds like it's 200 years ago when we think about the molecular world we currently live in. So, well, we talked about briefly the first time I think I heard you talk, but the first time we really interacted, we were meeting in the Clark building planning, I think Mike Snyder, who's now the ex-chair of genetics here at Stanford, but at the time was about to be the next chair of genetics. And I believe I remember well, actually, wandering into your office after I found it, and there you are, like pecking on your keyboard. I still think you don't do full typing, right? Of your many skills, that's not one. But anyway, I found you behind piles of journals. I still remember this. And we were supposed to talk about the seminar that we were putting together for Steve, but instead you had up on your screen your genome, which was just mind-blowing to me, because there were only, I think, 4 people in the world who'd had their genome sequenced. They were essentially mostly unknown other than Jim Watson. And you had sequenced your own genome with 2 people in your lab for, if I recall this, $50,000 or so on the machine that you invented, Helicos. And the one prior, I think, was done on Illumina with a group of like 30 people over 6 months. And, it just was a very different thing. It cost $250,000. So you'd already made headlines for sequencing your genome. But nobody had really spent a lot of time yet thinking about what medicine might do with that genetic information. And that was the beginning of an adventure, I think, a little bit of an adventure for both of us, certainly for me, and thinking about trying to build for ourselves first, for you, definitely as patient 0, and then for the world of like, what does it look like if you have an entire genome of an individual? And I tell that story a lot because it had a big impact on the way up my career went, but I wonder how you fit that into the overall picture of your career, where you've invented multiple things.
Well, there's a bunch to unpack there, I suppose.
Yeah.
When you go back to the early days of the Human Genome Project, when it was just getting underway, people appreciated the massive cost that it was going to take with existing technologies and the need for newer technologies. And so many of us in the physics community were aware of that. And thought, oh, we're physicists, we can invent measurement machines. And so a bunch of us were involved in that. I was one, and we, this is still, I was at Caltech, we invented the first single molecule sequencing and kind of published a paper on that. That turned into a company called Helicos, and they scaled it up massively. And, it was, their product was the first sequencer capable of doing an entire human genome with just one machine in principle, but nobody was actually doing it. So I'm like, all right, we're going to try it out, see if we can really make it happen. And so we did it. We sequenced my genome and published it. The editor at Nature Biotech wrote a really nice piece about how I was a genome altruist by giving my genome out there, because there were, at that point, every individual genome was revealing many new loci, hundreds of thousands of loci of human variation. And so it was really starting to fill out our view of what human variation looked like. And then we began to think about, can we interpret it? And you came in and we put a whole team together here at Stanford to try to answer the question of what happens when a patient walks into his doctor's office with a genome and says, help me. And, that was fun. And I loved being part of it because, as you may recall, I was the one who had done the sequencing, so I knew the strengths and weaknesses, potential errors, and how much trust you should have on any given base call. And so I was in the room while all the discussions were happening about what or what might not be in my genome, and the poor genetic counselor had her head in her hands. This was not the way it was supposed to happen in the textbooks, but it all turned out okay.
I think it did. But pretty interesting times, like you said. We were challenging our colleagues, genetic counselors, to suddenly deal with whole genomes and to think about how this might happen. You, as the inventor of the technology, were also the patient, and you were in the room as we discussed it, as you said. I mean, in the end, pretty clear at that point, really, no one had received medical advice on the basis of having their whole genome data. But we discovered a bunch of things with that big team, and it really took a team. I mean, Russ Altman helped, of course, on the pharmacogenomics side. Atul Butte's team really put together a whole engine for a common variant design.
And it was like an early version of a polygenic risk score, what he did, right?
Right,
I mean,
exactly.
like, very much ahead of its time.
Pulling together, built one of the first databases of genetic variation in relation to human disease, and then built polygenic risk scores. And then our team, we thought about rare disease, and we tried to put all of that together. And then also we had you in the clinic. Like, your family had been on at you to go see a cardiologist. there was one in front of you. So you ended up coming into our clinic, and
Yeah.
we tested you on a bike.
They took very good care of me.
Good. I'm glad to hear it. Yeah. Checked out your heart. Thankfully, we didn't find anything there, but we did find a few things that were of relevance for your disease and for your risk of disease, particularly heart disease. And I ended up making some of the first kind of recommendations that were genomically informed. I think famously, at least in my mind, we did an NPR interview together, kind of like this, as I recall. Might have been the last time you and I were next to each other in a microphone, and they asked if you had actually taken the advice that we'd given you, and I think at the time your answer was no.
You got me over the hump eventually.
Yeah.
You got me over the hump eventually.
Right. So glad to hear that you bought into the genomic medical revolution that you helped ignite. And now sequencing genomes for medicine is routine.
Yeah.
I mean, it literally is something that we do every day, and it happens around the world. So it's exciting, I think, to see that. I still think we're far short of realizing the full potential of genomic information for medicine. But especially for rare disease, I think we've seen a lot of lives impacted. And hopefully, I think that in addition to the work you did with cell-free DNA, that's a whole other area where I hope you would gain some sense of pride and fulfillment from being there at the beginning.
Oh, it was a ton of fun.
Yeah.
It was a ton of fun. And maybe to get back to your earlier question, once we did my genome, I felt like I had to make a decision. Did I want to get full into the human genomics field or do something else? And it was clear that the bigwigs in the field were just raising enormous amounts of money. And it was like for them all about scale and resources and how many could you do? And that didn't seem intellectually interesting to me.
Yeah.
And so, I kind of made a very conscious decision. All right, we did my genome. We did one tumor genome.
Yeah.
And I'm going to find something else to do.
Yeah.
And it was because of that decision and creating kind of the open space that I turned into liquid biopsies.
Yeah.
Right.
Yeah.
And that sort of, ended up being a very good decision.
Yeah. Well, and I remember at the time talking to you as we were thinking, we've done one patient, we've done 10. The next we have to do 100 or 1,000 and really think about how this is going to impact medicine more broadly. But at the time, it was around then that you had another conversation at Pete's Coffee, I think, with one of my forensic colleagues, Hannah Valantine. Just tell us about that.
Yeah, so we had published the first NIPT paper, and Hannah called me up and said, hey, is this going to work for heart transplant? Because we have a similar problem. We have to do invasive biopsies. And we've gone through all this trouble to give these patients a new heart. Right. Then what do? We go start pulling pieces of it out every couple of months to see if it's rejecting or not. Yeah. And wouldn't it be so much better if there was a blood test? Yeah. And so we put our heads together and figured out that we could monitor the specific cell-free DNA from that transplanted heart because the donor's genome was different than the recipient's, and the amount of donor DNA in the blood by actually sequencing and looking at polymorphisms would reflect how much rejection is happening or not.
Genome transplant.
Yeah, exactly. We called it a genome transplant.
Yeah. Well, and even more than that, I mean, I've done the cardiac biopsies, not for many years now, but I'm glad, thankfully, we're all doing many fewer of them. But the catheter that's used actually called a Stanford catheter, but that doesn't mean it's great. In this case, you're passing it through one of the valves, the tricuspid valve in the heart, and then just sort of poking it towards the middle of the heart, which, first of all, isn't necessarily the right place to go, but it's the place you can access. And then you just kind of grab and pull. You think of a biopsy, you hope that there's millimeter precision. This is not one of those things. It's rather a barbaric procedure, actually. So, the same way that amniocentesis is something that you would never invent if there was an alternative, I think that you could say the same for cardiac biopsy. So, but this was another story that, and you alluded to this earlier, it didn't immediately work. Like there were challenges with signal, but eventually it worked very successfully.
Yeah, that's right. I mean, the first pilot experiment worked very well. And then the first time we tried to scale it up to a larger study, it stopped working. And, it took a long time to get to the bottom of it. And, one of the interesting things about the project is it's a precision measurement. We're trying to measure things at the 1% level and, small sources of noise and error throughout the whole thing can really mess that up.
And this is not like with NIPT, you're counting chromosomes and doesn't matter, as you mentioned, whether you'd separate the mother and the fetus. Here, you really have to, the point is separating.
Here the point is to separate. And in fact, so we would genotype the donor and the recipient.
Yeah.
And some of the errors in the genotyping arrays were propagating their way through the whole thing.
Yeah.
And we had to figure those out,
Yeah.
compensate for them, and then it all started working.
Yeah.
But it took a while to get there.
Yeah. Yeah. None of these things happen overnight. But then a pivotal clinical trial, and then a company. And now, thankfully, there are many, many fewer cardiac biopsies.
Oh, yeah.
It's not just the heart, I guess.
No,
Kidney as well.
kidney, lung.
Yeah.
Yeah, it's a principle that works on any solid organ, which is super powerful and flexible.
Yeah, amazing. And of course, there's a whole revolution also happening. You talked a little bit about it today within the cancer world, thinking about both early detection and potentially multi-cancer early detection. And I think what is more, sort of embedded now clearly in mainstream medicine is the idea of minimal residual disease testing for cancer recurrence, let's say after surgery, which is very powerful. So it's moved towards molecular tests rather than relying on imaging. Why wait till a cancer is big enough that you can image it if you can pick up small numbers of molecules in the circulation? So it's really, I think, been extraordinarily impactful across all of medicine. Mostly been DNA, but RNA's out there too. Mm-hmm. And you have at least one company now that's focused on RNA and a lot of really interesting science. Just tell us a little about that.
Yeah, so cell-free RNA ends up being a very powerful way to measure phenotype. Not everything is genetic, and you don't always have a different genome in your body.
Right.
And so RNA ends up being a very good complement to DNA. And we initially went back to maternal health to try to solve the problem of preterm birth, which the genetic component contributor is very small, but there's clearly a phenotype thing. So we thought maybe RNA would provide a signal for that. And it does.
And the RNA is it specific cell types that it's reading out? Or what is the signal actually coming from?
Some of it comes from placenta, some comes from mom's immune system. Those are the two main contributors for preeclampsia anyways, and for preterm birth. You can also measure fetal transcripts, which is very interesting, I think, sort of scientifically. Yeah. But we haven't seen a clinical application of that yet.
Yeah.
And so that now led to another adventure. MIRV launched the Encompass test, 9,000 woman prospective clinical trial. Yeah. And so last year, that's now kind of out there, and it's been sort of amazing to see. Yeah. And for preterm birth, which is almost 10% of pregnancies affected by it, with no meaningful diagnostic. Yeah. Before this, we think that's going to be just another major impact. On helping save lives.
Any of us who've worked at the bench understand that DNA is fairly robust. RNA falls apart just by looking at it. It's pretty stable circulating.
It's a sort of counterintuitive thing that, the cell-free RNA that survives in your blood has been either trapped in exosomes or wrapped on protein particles. So it's got some kind of protection that has kept it intact. And it's remarkably stable, much more stable than completely purified RNA that you get on the benchtop, which is very labile. Yeah. And we've got amazing luck going back into banked plasma and serum from years ago, and lots of cell-free RNA. Because of it's not completely purified.
It's stabilized.
As the stabilized.
I think I remember one of the early insights I think from you or Dennis was just how short the half-life of DNA was in a pregnant.
Dennis did beautiful work on that. Absolutely. Looking at Y chromosome after delivery of a male baby, and
it goes down in
turnover time is half an hour. Right.
Half really short. Yeah. But the RNA is stable enough that even in bank samples you can find it. Yeah, that's remarkable. You've invented so many things. In the last few years, you've spent quite a lot of your time not taking grant money and inventing things with it, but rather giving out money with a big vision as part of the roles. You've played multiple roles within the Chan Zuckerberg Initiative. How did that first come about? And tell us about your time there.
Yeah, that was an amazing adventure. Yeah. When Mark and Priscilla decided to launch their science philanthropy, they reached out to the leaders of Stanford and UCSF and asked for advice. And the universities put together committees to help propose ways to design an institute and what to work on. I was part of that. Yeah. And then eventually got asked, along with Joe DeRisi, to be the founding leader of the institute, the first Biohub. And so we brought Berkeley into the fold. So we had all three major Bay Area institutions to take on a couple of big problems. One in infectious disease, which was Joe's interest area, and another in building cell atlases, which is what I wanted to do. And, you know, we were talking about this earlier, that powerful and as much as we know about the genome, you still can't predict the cell types of an organism from the genome. That's an unsolved problem. And we had developed single cell techniques to do that, to characterize gene expression of single cells, and thereby, in principle, create an atlas of all the cell types in the human body. And we set out to do that at the Biohub, and succeeded. You know, it was just great team science. There's like 150 authors on each paper, and involved dozens of groups from all three institutions, and figure out how to coordinate that was a new thing for me.
You were getting every human tissue and then sending it out to labs and then getting
a lot of work
single cell level, right? And then that is available to the world.
We just gave the data away, absolutely, even before we published it. So we had a very strong commitment to open science, and we were probably the first organization to require the use of preprints. So everyone we funded, we said, you've got to put a preprint of your work up when you submit it to a peer-reviewed journal. And for our big projects, we not only put the preprint up, we put the data out there. And we gave people access to the data before the papers were published, and in the name of trying to accelerate science and lift boats everywhere.
I mean, I think there isn't an institute or initiative with a more bold aim. This is obviously something that people focused on, managing or curing all disease within, I think, initially a 10-year period, and maybe a little bit longer.
100-year period.
100-year
period.
Okay.
Yes.
Okay. And I think that the word manage is key, but we live in an amazing moment for discovery science and its translation to medicine. And I think, this was your initial work in the context of Biohub. The last few years, you were also helping build other Biohubs. There's multiple of them now.
Yes, exactly. Well, I moved on to manage all of Mark and Priscilla's science philanthropy as the head of science. And part of that was creating a Biohub network, which we established in Chicago and New York, as well as in collaboration with the Allen Institute and Outpost in Seattle. And more generally, funding science globally. I mean, we funded grants in something like 30 different countries. And that was a lot of fun to be part of that, and to really try to get that launched. And part of what I got to do was lead the pivot towards AI. Right. I mean, that was, not the obvious thing to do at the time, but it was, sort of, there was enough early kind of examples that it felt like the perfect thing for CZI to be investing in. It's still risky, wasn't clear it was going to work out, but a good thing for philanthropy to do.
What struck me with that was just how elegant the foundational work for the cell atlas is for the pivot towards AI, the idea of AI in biology. I mean, and it couldn't have been predetermined, because so much of what we understand about the power of the new AI in this third revolution is only been 2 or 3 years old. So in a sense, you might have had other reasons for originally initiating the cell atlas work, but of course it was just good for the world, and then it's perfectly poised. Thinking about now a sort of in silico immune system, or some of these other ideas that are coming out as potential priorities for the Biohub group, Biohub network going forward. I think Priscilla's actually coming on our podcast and to our grand rounds in a month or two, so we'll be able to follow up and ask her.
Is she going to give grand rounds herself?
I think she might. We may do it in this format, though.
Right.
We may talk about it.
She being a physician, she's certainly qualified to.
No, we're very excited that they have such a big vision that goes all the way from very basic discovery all the way through to disease. And obviously, we're really excited about the impact of AI at multiple levels, molecular levels, cellular levels, all the way up to patient interaction levels. So we have missed out a whole bunch of stuff that you've invented over the years and impact that you've had. But I wanted just to spend the last few minutes sort of thinking about more generally, in anyone's early career, physician scientists, they're trying to build, hoping for maybe one big hit, something that has impact, maybe all the way through to a patient, and maybe starting a company that has some impact. You've done this tenfold more than most people, and I wonder what lessons do you take, or what do you ascribe that sort of disproportionate success?
It's hard to know because I don't get to replay the tape. part of it is being willing to jump into the unknown,
Uh-huh.
and work on emerging areas rather than mature ones.
Yeah.
And that enriches your chances, I think, of
Yeah.
doing something like that. And then, so I've transitioned in my career from one area to another. And as, I sort of, when things get mature, I get bored.
You're right.
And,
Right.
I look for sort of what's the next emerging thing to work
Where's the
on.
gunpowder?
Yeah, exactly. Yeah. I mean, also, it's just working with terrific people. I mean, I've been so fortunate to have great students and postdocs and collaborators over the years, and that's driven so many things as well.
We've talked a little about in the past about the sort of secret sauce of Stanford and the ready collaboration, but also just the fact we're all here in one place. Is that, do you think, an important part of?
100%.
Yeah.
When I moved from Caltech to Stanford,
Yeah.
I knew that there were a bunch of cell biologists and such forth that I wanted to collaborate with, the stem cell folks and that all came to pass.
Yeah.
I had no idea how much fun I'd have with the doctors and
Yeah.
physicians, right? That opened up a whole new world to me. And
Yeah.
we've done things, as we talked about in the grant round. That's the whole story of that,
Yeah.
right?
Yeah.
Over the last 20 years, that I never thought that was going to happen. And
Yeah.
it would have been hard to do it anywhere else.
Right. It is a great environment for that. Last question then really is, I mean, this podcast is called The Future of Medicine, right? There are few people better positioned. We've certainly had a lot of guests on who've given us their view. Eric Topol was on recently, and he's very well positioned as well. But you're someone who invents the future. So what are you most excited about, the next few years, either for yourself or just more generally?
So I am on a sort of liquid biopsy world,
Yeah.
very bullish about the early cancer detection work, multi-cancer detection. I feel like there's been a bunch of early attempts, a bunch of marketing and things.
Yeah.
It hasn't quite come together yet,
Right.
but it's poised to do it now.
Yeah.
I think we know enough collectively
Yeah.
that that's going to really...
And do you think that will be DNA methylation plus RNA? Do you do you have a sense of what what the measurement will be of?
I'm most optimistic about hydroxymethylation.
Oh, okay.
That's like your best compromise between methylation and RNA. It's right in between.
Right.
It's the genes that are being turned on, but it's a DNA measurement. It's remarkably predictive. The NCI is doing this Vanguard trial of early cancer detection. There's only 2 companies in it, and one's methylation, the other's hydroxymethylation.
Right.
But the other one I'm super enthusiastic about is dementia
Yeah.
and neurodegeneration. I think, there's going to be tests for that. That whole field is ripe for disruption.
Yeah.
There's been a lot of orthodoxy and received wisdom, and
Yeah.
I think there's going to be some amazing things happening there.
We certainly need it. Well, Steve, we could talk for much longer, and you've certainly done many more things, but I'm glad to have a chance to talk about your entire life so far. Many more things still to happen. Thank you for joining us on The Future of Medicine.
Thank you, Ewan. It's been great.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.